In this video, we at the Nathan Lab would like to show you step-by-step how we create differentially detectable Mycobacterium tuberculosis. When working with Mycobacterium tuberculosis, all the following steps should be performed in a biosafety level 3 lab. Before beginning, please review these two slides regarding the materials that you'll need and how we prepare MTB cultures. This section will describe how we wash and resuspend the culture in phosphate-buffered saline. First, collect the mid-log phase cells into a 50 ml falcon tube. Note that whenever we collect cells, we wash the sides down to maximize the yield. The culture is spun at 3082 times G for 8 minutes with maximum acceleration and maximum deceleration. The supernatant is discarded, and the pellet is first resuspended with 1 milliliter of PBS with taloxipol. After vigorous resuspension, a volume equal to that of the original culture volume is then added. This washing with PBS with taloxipol is done twice. After the second wash and resuspension, a single cell suspension is formed by spinning at 123 times G for 8 minutes with no deceleration. The supernatant is collected, taking care not to touch the pellet. The OD of the supernatant is then checked. Enough PBS with taloxipol is then added to the single cell suspension to dilute to an OD of 0.1. A small volume is set aside for CFU of the inoculum, and the remainder of the single cell suspension is divided equally among 3 vented flasks, which are then placed at 37 degrees Celsius with 5% CO2 for 2 weeks. In this section, we will add compound to the PBS exposed cultures. After the PBS exposure period, the culture is mixed via pipette. At least 100 microliters of culture is set aside in a microcentrifuge tube for MPN dilution and CFU, which are shown in detail in the last section. Each triplicate culture is then divided into equal volumes for every compound exposure arm and DMSO control arm. Next, drug diluted in DMSO, or DMSO control, is added in triplicates, taking care to keep DMSO percentage the same in all arms of the experiment. These vented flasks are then incubated for 5 days at 37 degrees Celsius with 5% CO2. This section will describe how to wash the cells after the exposure period. First, the cells are collected and spun at 3082 times G for 8 minutes. The supernatant is then discarded. The washing process begins with vigorous resuspension in 1 ml of PBS with taloxipol. PBS with taloxipol is added in a volume at least 5 times greater than the initial culture volume. The cells are again spun at 3082 times G for 8 minutes. After the spin, the supernatant is again discarded. However, this time we use a 2-step process in order to maximize the amount of supernatant removed. First, the bulk is removed with a large pipette. Then, a second 5 ml pipette is used to remove supernatant until about 500 microliter remains. This removes the bulk of supernatant without disturbing the pellet, even if one is not visualized. The pellet is resuspended in the remaining fluid and transferred to a microcentrifuge tube. During the transfer, the volume is noted. The original falcon tube is then washed with the amount of PBS with taloxypol needed to make the entire final volume 1 ml when added to the microcentrifuge tube. Note that this creates a higher concentration than the original culture and must be accounted for in later calculations. This sample is then used in the MPN dilution. This section will describe in detail the MPN dilution process. First, using a sterile trough, fill all wells of a 96 well plate with 135 microliters of 7H9 supplemented with glycerol, OADC, and taloxypol. Next, vortex the sample to be added 3 times, 10 seconds each. Note that when this is done with live MTB, the vortexing is done within the biosafety cabinet. Add 15 microliters of sample to column 2, rows B through F. Prior to continuing to the next step, prepare a Vespine trough and a tip waste container. Using a multi-channel pipette set at 100 microliters, pipette up and down 10 times to mix the 5 wells. Note also the slight rotational component to ensure complete mixing. Dip the tips into the Vespine trough prior to discarding into the tip waste container. Next, use another multi-channel pipette to transfer 15 microliters from column 2 to column 3. Repeat the process of mixing and transferring to create dilutions up until column 11. After mixing column 11, again take up 15 microliters, but discard this amount of sample so that all columns have equal volumes. When the dilutions are completed, use a multi-channel pipette to transfer 35 microliters from each row to a single row of a new 96 well plate, combining the rows but preserving the dilutions. This produces a final volume of 100 microliters in the MPN plate. Use this new 96 well plate for CFU. Note that the wells used for CFU are mixed again just prior to plating. We use 7H10 tri-plates in order to fit 3 dilutions on one plate. 10 microliters of the appropriate dilution is placed in each section of the tri-plate. Then, the droplet is spread evenly using an inoculum loop. In the last step, we use a new sterile trough and add 100 microliters of supplemental 7H9 to all 5 rows of the MPN plates. Note that culture filtrate or other addition can be made to the MPN plate instead at this step. Both before and after adding to the MPN plates, we keep an additional plate of just supplement as a control for contamination. Plates are then taped together in stacks of no greater than 4 and kept in an open Ziploc bag. Incubate at 37 degrees Celsius and read at 3 and 5 weeks. It is advisable to keep the plates as long as necessary until subsequent reads do not produce any further difference. Plates are read by visual inspection using a black backdrop. Each well is a binary output, no growth versus any visible growth. When reading, the results are tallied onto a sheet prior to inputting into the MPN calculator. Note that the dilution factor and the volume of the dilution are recorded in the MPN calculator.